In this lecture, we're going to talk more about the big pictures of regulation on a cellular level. The feedback inhibition example that we just talked about in the previous lecture is an example of global regulation. One can think of basic regulation as one regulator affecting one target. Each pathway also has to take into account the global state of the cell and act accordingly. There are other circuits where the regulators affect more than one pathway, especially in a global sense. Genes are often not adjacent on the chromosome. These sets of genes that can be affected by a single protein are called regulons and are common in bacteria. The table lists some examples of regulons in E. coli. You'll see that there's one for aerobic respiration, anaerobic respiration, catabolite repression, etc. To drive this point home, let's spend a few minutes on three systems, looking at them from a global perspective. These are catabolite repression, heat shock, and quorum sensing. In addition to the inducible repressor system described previously for the lac operon, it is also under positive control by a system that senses carbon levels in the cell. E. coli is capable of growth on numerous different carbon sources: glucose, lactose, galactose, maltose, rhamnose, and arabinose, to name a few. Remember that glucose is the energetically preferred carbon source, with the others, such as lactose, being less desirable. If glucose is present in the environment, it would be inefficient and a competitive disadvantage to express the genes for utilization of any of the other sugars, even if those sugars are also present. To solve this problem, the cell has a regulatory mechanism to prevent the expression of these other catabolic enzymes in the presence of glucose, called catabolite repression. This circuit must work in concert with the specific regulatory proteins that control the expression of each of these catabolic pathways. In the presence of glucose, these pathways must be turned off, irrespective of the presence of these other carbon sources. However, when the secondary sugar is the only available carbon source, the pathway should be turned on. In this way, the cell is able to globally regulate the expression of a number of operons based upon the overall carbon and energy state of the cell. There are three players in the global control circuit: the phosphotransferase system, PTS, that transports glucose into the cell, adenylate cyclase, and cyclic AMP receptor protein. The important protein of the PTS for catabolite repression is named 2A glucose. This protein undergoes phosphorylation during the transport of glucose. If glucose is present, 2A glucose adds phosphate to glucose and is not phosphorylated for long. However, if glucose is not present, then 2A glucose remains phosphorylated. In this phosphorylated state, it binds to adenylate cyclase, thus activating it. When activated, adenylate cyclase catalyzes the conversion of ATP to cyclic AMP. This synthesis of cyclic AMP is then a signal molecule that indicates that glucose is not present in the cell. CRP is the regulatory protein that senses cyclic AMP. It is synthesized in an inactive state, and binding of cyclic AMP causes a conformational change in CRP that enables it to bind to specific sites on DNA. CRP is a positive regulator and binds at a number of sites, including just before the lac promoter. It recruits RNA polymerase to the promoter, and if lactose is present, it allows transcription. If CRP is not active, meaning that there is glucose in the cell, it will not bind to the operon, and it will not recruit RNA polymerase. Even if lactose is there, it's going to not efficiently recruit RNA polymerase because the CRP protein won't be there. Let's check your understanding. Under what conditions would expression of the lac operon be highest? The correct answer is B, no glucose plus lactose. Remember, glucose must be absent so that the CRP cyclic AMP binds to the operon, and lactose must be present so that allolactose is made and inactivates the lac repressor. That is the only time you'll get significant transcription of the lac operon. Before we talk about heat shock, I want to introduce one or more regulatory paradigm. If the cells need to change the expression of a large set of genes at the same time, it will sometimes use an alternative sigma factor. Instead of having a regulatory site that a protein binds to, a sigma factor is made that can direct RNA polymerase to a different promoter sequence. All the genes involved in the process will have an alternative promoter that the sigma factor recognizes. E. coli has 7 different sigma factors, and they're shown in the table here. And you can see that each sigma factor has a different upstream recognition sequence for its promoter. There are sigma factors, a vegetative sigma factor, one for nitrogen, one for when you enter stationary phase, the heat shock response, which we're going to talk about now, one for flagellosynthesis, one for dealing with misfolded proteins, and finally one for certain genes involved in iron transport. This is a common phenomenon that other bacteria contain multiple sigma factors too. Syncellocystis, a cyanobacteria, has 9, Bacillus subtilis, an endospore former, has 18, and Streptomyces colicolor has 63 in its DNA. Now, there's also anti-sigma factors that inhibit specific sigma factors, but we won't be talking about those in this class. The heat shock response is our next example of global regulation. All living systems respond to environmental stress, including heat. The heat shock response prepares a cell to survive adverse temperatures. There are several categories of heat shock proteins. There's regulators that control the heat shock response. There's molecular chaperones that are involved in helping proteins fold during their synthesis. And there are proteases that are produced to degrade misfolded proteins that cannot be rescued. HSP, or heat shock proteins, are present under all circumstances, since the cell always needs protein folding functions and needs to dispose of proteins that are misfolded. But levels increase at high temperature. HSPs are also induced under other stress conditions, such as ethanol, too much ethanol. The master regulator of the system in E. coli is sigma H, also called sigma 32. The overall regulation of this response is as follows. Immediately after a temperature increase, the rate of synthesis of the chaperone proteins increases fairly dramatically until the level of these proteins reaches the level of accumulation appropriate for the higher temperature. At that point, the rate of synthesis slows somewhat, but to a new steady state level higher than it was before the temperature increase. This higher rate of synthesis is necessary to deal with the elevated demands on heat shock proteins at the higher temperature. The cornerstone of this regulation is RPOH, which recognizes promoters for all the heat shock genes. During a heat shock, the concentration of RPOH increases several-fold and thus increases the expression and subsequent concentration of all the heat shock proteins. Part of this increase in RPOH levels is due to an increase in RPOH transcription, but significant post-transcriptional regulation occurs in 2 places, at the level of mRNA secondary structure and at the level of protein stability. The secondary structure of mRNA can play a role in the amount of translation that occurs because the accessibility of the ribosome binding site affects the frequency of ribosome binding and translation initiation. This process is rarely regulated by external factors, but one of the best known examples of such regulation is the mRNA of the RPOH gene of E. coli. The mRNA of RPOH has extensive regions of secondary structure that mask the Shine-Dalgarno sequence, making it more difficult for the ribosome to bind to it. During a heat shock, however, the rise in temperature causes the secondary structure to melt, allowing the ribosome to bind more readily. This then causes a 5 to 10-fold increase in the amount of translation and a rapid rise in the amount of RPOH. The stability of RPOH also regulates its concentration in the cell. During vegetative growth, RPOH is constantly synthesized, but is highly unstable, having a half-life of only a few minutes at 30 degrees centigrade. Such instability is unusual for proteins, and in this case is a deliberate result of the action of three proteins, DNA KJ, GRO-ELS, and FTSH. DNA KJ and GRO-ELS are chaperones that also bind to RPOH. When bound, they hold RPOH in a conformation that makes it susceptible to degradation by the FTSH protein. At 30 degrees, DNA KJ and GRO-ELS are in excess, and enough is available to bind to RPOH and keep its concentration low, resulting in a relatively low level of heat shock proteins. When cells are shifted to higher temperatures, DNA KJ and GRO-ELS recognize the presence of the many misfolded proteins, and therefore they carry out their basic function, which is to help refold these proteins back into functional proteins. That's what they do. They're molecular chaperones that help in protein folding. When they're doing this, they're too busy to bind RPOH. This greatly increases the half-life of RPOH and thus its effective concentration. A higher concentration of RPOH leads to greater transcription of heat shock promoters, one of which is present in front of DNA K and DNA GRO-ELS. This also causes a rise in DNA KJ and GRU-ELS levels, themselves heat shock proteins, which eventually reach a level at which they can again bind up all the RPOH and degrade it. As the level of RPOH decreases, so does the amount of expression of the heat shock genes. This is a beautiful example of the cell regulating the level of proteins, DNA KJ and GRO-ELS, in this case, after testing directly if their level of function was appropriate for current cell conditions. Let's see if you've got that. If you remove the secondary structure of the RPOH gene, but did not change the actual codons, expression of the heat shock genes would... The correct answer is increase. Without the secondary structure, the mRNA of RPOH would be translated more quickly, leading to more protein. Since there is more RPOH, there will be more transcription of the heat shock genes. Okay, now we move on to our last subject in regulation and also talking about global regulation, and that is quorum sensing systems. Before I talk about quorum sensing systems, however, I wanted to explain two-component regulators. Sometimes the signal is outside and you don't want to transport it inside, or you just don't need to transport it inside. And so the question is, how do you get that signal into the interior? One of the solutions that bacteria have come up with is two-component regulators. And this is a solution that's used in lots of different microorganisms and bacteria. First, there is a sensor kinase. And this is an enzyme that's in the membrane, and it will bind the environmental signal, and this will cause a change that causes the sensor kinase to phosphorylate itself. It then transfers that phosphate to a response regulator. This response regulator will often have an activity that involves it binding to DNA, and it will either turn on or turn off expression of genes. It just depends, and you'll see specific examples as we go through quorum sensing. This response regulator binds and then has its effect. At some rate, there is a phosphatase. Sometimes this phosphatase is part of the response regulator, sometimes it's a separate enzyme that will clip off this phosphate, and then the response regulator loses its activity. So it will only stay on if it continually gets rephosphorylated by the sensor kinase. Thus, when the signal goes away, the phosphorylation of the response regulator will gradually go away, and then the response changes. Now, I do want to say that response regulators are not always proteins that will regulate transcription. Sometimes they do something else. Second, so the sensor kinase is not always in the membrane. There's examples of sensor kinases that work in the cytoplasm. Two-component systems are present in all three domains, and they're very common in bacteria. E. coli itself has about 50 different systems. And here is just an example of some of the systems that are in E. coli. I don't expect you to memorize them all. Right, so now we come to quorum sensing. Quorum sensing is the ability of microorganisms to sense how many of them are in the environment. And we're going to go through two examples of this because it seems to divide into two general patterns. One for gram-positive bacteria and one for gram-negative bacteria. In general, these systems are used to sense what's going on in the environment and how many of them there are. You secrete a small molecule into the environment, and as the concentration of that small molecule increases because there's more of the bacteria secreting the molecule into a confined space, it indicates that a population is larger, and then when that happens and the concentration is high, then it affects gene expression. And as I said, there's all sorts of examples of quorum sensing systems, and a lot of them are listed here. I mean, you see them in bacteria, you see them in yeast. One system is the AGR quorum sensing system, and this is going to be our example of what happens in gram-positive bacteria. AGR stands for accessory gene regulation, and this is a system in Staphylococcus aureus. Staphylococcus aureus, as you find out later in the semester, is a really important pathogen. And this is the system it uses to sense if it's at high number. And often what that means is it's infecting a host. And therefore, the end result of this process is it turns on genes that are important for virulence. Let's look at the enzymes and proteins that are involved in this process. AGRD is the gene that encodes a pre-pro peptide that's processed into the auto-inducing peptide. This is the small molecule that Staph aureus senses in the environment and then acts accordingly. AGRA and AGRC are a two-component system. AGRC is the sensor kinase. AGRA is the response regulator. This causes the synthesis of RNA3, and then RNA3 causes the regulation of all sorts of other genes. Let's look at this. So promoter 2 here transcribes all the genes here. AGRB, AGRD, AGRC, and AGRA. AGRD is the pre-pro peptide. It comes to the membrane. AGRB, along with SPSB, then go here, and this is clipped. This is transported across the membrane and processed into the auto-inducing peptide. Now, if the cell is at low concentration, this auto-inducing peptide just diffuses into the environment and nothing happens. However, if the bacteria gets into an enclosed space and the number of Staph aureus increase, then the concentration of AIP increases. Some of it starts binding to AGRC. That will cause this to activate phosphorylate AGRA, and then AGRA sits down at this region and is an activator, and it will turn on transcription of not only RNA2, which is the genes for the operon, it will also turn on transcription of RNA3. RNA3 is a RNA that then regulates a whole bunch of genes. It is an antisense RNA. It activates translation by base pairing and leaving a stem loop that masks a Shine-Delgarno sequence. In other transcripts, mRNA transcripts, it inhibits translation by base pairing with the Shine-Delgarno region and occluding it from the ribosome or making it a target for ribonucleases. Again, this is expressed when there's high populations of Staph aureus. Okay, so here's two examples. The HLA gene, which encodes alpha toxin, normally has its mRNA and it will form a stem loop that blocks its Shine-Delgarno sequence, so it will not be translated when it's just made natively. However, when RNA3 is made, shown here in red, it will bind to this upstream 5' region, block it from binding to the Shine-Delgado region, and now it's open for translation and it turns on. Thus, the presence of RNA turns on the expression of alpha toxin, which is a known
virulence factor for this bacterium.
It can also have a negative effect. The SPA which is gene which encodes protein A, has its two stem loops like this, and its Shine-Delgado sequence is open and available for the ribosome, so it will be translated. However, if RNA3 is made, this antisense RNA will bind to this region, it will block the Shine-Delgado sequence, and it will prevent translation. In addition, this binding creates RNase sites that a specific RNase recognizes and will degrade the messenger RNA. So not only does it block expression of translation of the Shine-Delgado sequence, it also causes the degradation of the RNA. So in this case, the presence of RNA3 blocks translation. Finally, RNA3 binds the Shine-Delgado region of ROT mRNA and blocks its translation. The ROT protein is a negative regulator of many virulence genes. So by expressing RNA3, it stops a negative regulator of virulence genes, and thus they all get turned on. So just to summarize, the mechanisms involved in Staph aureus are, first of all, quorum sensing. You have two component regulators, and then you have an antisense regulatory RNA in the form of RNA3. The outcomes of the AGR regulation are the AGR system is off in log phase and at lower pH, and that may be, quote, normal, unquote, conditions for Staphylococcus aureus.
The expression of the AGR system increases in stationary phase or elevated pH.
AIP increases in the concentration of laboratory culture. This then ultimately leads to the increase in RNA3 expression. RNA3 then affects the expression of many virulence genes. Multiple toxin genes are turned on. Secreted proteases, fatty acid degradation systems, immune system interfering proteins, and antiphagocytic capsule synthesis all get turned on in the presence of it. So here is the clicker question. If you mutated AGRB so that it could transport but not degrade AGRD, so not clip it and make the auto-inducing peptide, would the AGR system still work for quorum sensing? We now come to our last topic in regulation, and that is quorum sensing in gram-negative bacteria. I hope you watched the video by Bonnie Bassler, who is just an amazing person and has done great research at Princeton. So definitely go look at that. And it's a TED Talk, so she explains it more for the layperson, but it's definitely something that's worth watching. This was one of the first systems ever discovered, and this is yet another example how sometimes the esoteric interests of scientists that seems to have no relation or importance can pay off in huge dividends. Once we learned more about this system, we discovered it was present in all sorts of microorganisms, some of them important pathogens, and has now led to avenues of investigation that may be able to stop those pathogens. Let's look at the system. There is a host microbe symbiosis between Vibrio fischeri and a Euprymna scalopus. Euprymna scalopus is the Hawaiian squid, and it takes up bacteria into its light organ. The bacteria then bioluminesce in the light organ of this squid, and this light provides protection for the squid because it's a night feeder. Now you may think that doesn't make a lot of sense. If you're an organism that swims around in the dark, why would you want to glow in the dark? You think then predators would see you. Well, it turns out that the squid swims in the ocean, and it's normally above the predators that will eat it, and they will be looking up for it from below. On nights when the moon is shining, the squid would look as a blotch, a dark blotch in a light background. But if the squid bioluminesces, then it blends into the moonlight and you can't see it anymore. Amazingly, the squid is smart enough that on nights when it's cloudy, it won't take up Vibrio fischeri. It will block the bioluminescence, and it then is dark in a dark background. So the light provides protection for the squid. The bacteria grow and bioluminesce, and they gain what most microorganisms gain in these mutualistic relationships: a nice home and free food. Right? They gain sugars, amino acids, peptides, and proteins. This light organ of the squid is highly selective. It's basically a pure culture of Vibrio fischeri. So let's look a little bit at the biochemistry of this. There are 6 genes involved in the actual biochemistry. The main genes that cause the light are luciferase, and that is a dimer of the LuxAB genes. They reduce a long chain aldehyde with electrons. This consumes oxygen and produces light. The LuxCDE proteins are involved in taking things out of fatty acid biosynthesis, these long fatty acids, right, shown right here, and then changing them to an aldehyde. And that aldehyde is now ready for processing by the luciferase. The LuxG gene reduces flammimononucleotides so that it can then participate in this reaction. So that's what the 6 genes do. And then here's the luxoperon. You can see that it's 2 transcripts: the LuxR protein, which is a regulator of the system, the LuxI protein that synthesizes the autoinducer, and then the structural genes of the operon. In autoinduction, if you are at low conditions, the LuxR protein is synthesized and released into the cytoplasm. Right? So the LuxR protein is here. This is an activator, not an inhibitor. It is an activator of transcription. The autoinducer LuxI is synthesized, and the LuxI protein synthesizes the autoinducer. So this is an enzyme that produces the autoinducer. This autoinducer is membrane permeable, so it just diffuses out of the cytoplasm into the environment. Under low concentrations, again, this diffuses away and nothing happens, as shown here. Under low cell populations, there's low LuxI made. That means it doesn't make a lot of the autoinducer. Again, the LuxI protein is an enzyme that synthesizes the autoinducer, homoserine lactone. Therefore, none of it binds to LuxR, and there's no transcription. Under high cell populations, LuxI then synthesizes the autoinducer, and since there's lots of Vibrio fischeri, there's lots of this in the environment, it diffuses in and out of the cells. It then binds to LuxR, is a activator, right? It sits down on the DNA and it activates transcription. Transcription turns on, you make more LuxI, you make more autoinducer, the concentration increases, and then you get even more of this system made. Okay, finally some active learning exercises. A mutation is made in the LuxI protein such that its activity is 10 times that of wild type. In other words, the enzyme is 10 times more efficient, makes 10 times as much autoinducer than it normally would. In this case, strains of V. fischeri would bioluminesce, and there's your choices. Okay, finally, one more thing that I suggest you practice is to summarize quorum sensing. And the best way to do this is compare quorum sensing in S. aureus to that found in Vibrio fischeri. What do they have in common, and what is different? Okay, that brings us to the end of the lecture on regulation of metabolism. Our next lecture will be on genetics and genomics.